Yeah. So thank you very much, Caroline. So now we move to the next presentation by Jennifer Grossman from the US, from the NCI-NCL in the US.
Hi, my name is Jennifer Grossman. I'm a physical chemist and a senior scientist at the US Nanotechnology Characterization Lab, which is located in Frederick, Maryland. And today I'm going to talk a little bit about how we've been working through this transatlantic collaboration with the group that's trying to establish the EUNCL to try and accelerate that establishment. And I think Caroline gave a great explanation of why you need a resource like the NCL or the EUNCL and the complexity involved in characterizing nanomedicines for the regulatory process. Hopefully I can explain how we've been trying to train the EU scientists to provide a similar service. So a little bit of background, although my colleague Scott McNeil gave an overview and my colleague Marina Dobrovolskaya, who's the head of our immunology group, gave an overview. But the NCL was founded in 2004 as a collaboration between the FDA, the National Cancer Institute, and the National Institutes of Standards and Technology. And the idea was that we would accelerate the translation of promising nanotech cancer products by providing a free service that would help meet regulatory requirements. And essentially anyone who's developing a nanomedicine product can apply to the NCL. They apply by demonstrating the efficacy of their product in comparison to currently available standards of care. If it's accepted, we provide what is essentially a free research and development program, a testing program, using assays that we developed that are specific for nanomaterials. And at the end of the collaboration, the data is provided as a package to the sponsor that they can use to advance their technology, to garner investment as part of the regulatory submission, however advances their technology. So this, very quickly, is our assay cascade of tests. It consists of physicochemical characterization, things like size and size distribution, in vitro characterization, in vitro biology, hematology, immunology. Marina covered some of this in her talk yesterday. And animal studies, in vivo characterization, we do non-GLP animal studies in rats and mice. And quickly, we have now characterized over 350 different nanomedicine formulations, and from the gamut of types of nanomedicines that are in preclinical and clinical development. So this is a pie chart of some of the types of nanomedicines that we've tested at the NCL. 10 of our collaborators now have products that are in clinical trials. And so providing this service since 2004, for 12 years, has given the NCL unique expertise. But there's no reason that we need to be unique, because our assay cascade is really geared at meeting the FDA's regulatory requirements. And we don't have a lot of expertise. We've gained some over the last year about the regulatory process and requirements in EU member states or in other countries. And so we were approached a while ago now to work with the EU to develop a transatlantic collaboration for the EUNCL. And the reason we signed up was that we thought that this collaboration would really expand the visibility of global nanomedicine. It's good to have a transatlantic collaboration between two different countries. We thought it would leverage our historical knowledge base and experience, and we thought that it would lead to better quality control across the board. If we have a group in the US of experts and a group in the EU, we're not duplicating efforts. We can reduce the risk of an adverse event in nanomedicine. And the hope is that it will facilitate regulatory convergence for nanomedicines. Since the NCL works with the FDA, the EUNCL can work with the EU national authorities and the EMA, and together, if we're also working together as the NCLs, we can agree on the science, and that can help coordinate or inform the regulatory process in both countries, which should lead to better coordination. So what have we been doing in the past year? We have a close collaboration for knowledge transfer. How do you transfer 12 years of experience from a group of nearly 30 scientists to a group of other scientists with different backgrounds? And what we've been doing is hosting guest researchers from the EUNCL, technical trainings on our methods, cross-validation, providing supplemental characterization capabilities, and conducting interlaboratory studies. I want to talk a little bit about the specifics. We had a 2-week training at the US NCL for 13 members of the EUNCL core expert team in December. The week 1 was technical training on NCL methodology, assay nuances and troubleshooting, selection of controls, which is very important for these assays. Week 2 was on operational workflow, quality management, very nitty-gritty in the details of how we assess samples and store samples and aliquot samples, but all of that is important to maintain quality. And we've had multiple follow-ups for training, discussion with our entire staff. This is to give you a flavor of the training. It was very intensive. We did drug loading, cytotoxicity, platelet aggregation on Tuesday, lipid composition assays, hemolysis, complement activation. We didn't have time to go through to perform all of these assays, but it was okay because the EUNCL experts had already familiarized themselves with them, already tested them out in their own laboratories, and we were really training them on troubleshooting and selection of controls. We've also been doing some cross-validation of the assays run in our lab with the assays run at the EUNCL labs. They started out testing research-grade liposomal doxorubicin, which we've done quite a bit of testing on in the NCL, and we compared results. Now underway is a comparison study using Onivide, which is a liposomal irinotecan that was approved in October in the United States for pancreatic cancer. And our lab is currently testing it. The EUNCL labs are also currently testing it. We expect to be done in September. We also did an interesting exercise using bugged samples with the knowledge that the pristine, already approved Onivide sample wouldn't have some of the issues or challenges that preclinical samples in early development have. So we thought about our experience and some of the challenges that we had faced and made trick samples, bugged samples for the EUNCL to test. And those had things like endotoxin contamination, an incorrect drug loading. We told them it was actually a different drug than the drug that the particles were loaded with, a composition that we didn't tell them about that they had to find, and cytotoxicity coming not from the nanoparticle, but from the buffer surrounding the nanoparticle. And I'm pleased to report that the EUNCL centers, all of them successfully detected the bugs. So far, the training has gone really well, and our team has been impressed with the quality of the expertise in the various EUNCL centers. So the future, we're looking forward to it. After the EUNCL is up and running, that's not going to be the end of our collaboration. We are expecting to continue to work with the operational EUNCL. We're looking to collaboratively develop novel methods to continue to conduct interlaboratory studies and do trends analysis. And we think that the US NCL and EU NCL can inform the FDA and EU regulatory science for nanomedicines and increase coordination and convergence between those requirements. And a complementary regulatory framework really does decrease perceived regulatory hurdles for these very complex nanomedicine products, and it should increase investment and enthusiasm for nanomedicine in general. And the other future of the US-EU EUNCL's collaboration is to continue to support and promote global nanomedicine R&D. Thanks.